Okay, ladies and gentlemen, good day all. My name is Mukhtar Aliyu. In today's lecture, we are going to look at the concept of chemistry of carbohydrates. So what is carbohydrate? Carbohydrate is defined as a polyhydroxyaldehydes or polyhydroxyketones or any compound that produces that on hydrolysis. So carbohydrate is defined as a polyhydroxyaldehyde or polyhydroxyketone or any compound that produces such an hydrolysis. So the most important thing that you should put at the back of your mind whenever we actually talk about carbohydrates, they have two main functional groups. They either have aldehyde functional group. Remember from the basic knowledge of your chemistry. In organic chemistry, we have what you call a carbonyl compound. When we say carbonyl compound, it means that they are compounds that have carbon to oxygen double bond. So they are compounds that have carbon to oxygen bond, and they are called carbonyl compound. So therefore, these aldehydes and ketones, they are a typical example of carbonyl compound. And they are, of course, the family of organic compound, and they have carbon to oxygen double bond. So carbohydrate is defined as a polyhydroxyaldehyde. It means that there must be aldehyde or ketone functional group in carbohydrate. So, but now the question is, what is the difference between aldehydes and ketone functional group? So the difference between aldehyde and ketone functional group is simple. In the case of aldehyde functional group, this is a carbonyl carbon. So in the carbonyl carbon, there is free hydrogen. There is free hydrogen here. And of course, there is alkyl group here in the case of aldehyde. So these are stand for aldehyde functional group. So it has a free, actually in the case of aldehyde functional group, there is a free hydrogen that attached to the carbonyl carbon and the R. This R stands for alkyl group. So this is R. Although it is not clearly written. So this is of course R. It is R. So now, of course, what is the basic differences between the aldehyde and ketone? So in the case of aldehyde, there is R here. And then, of course, in the case of the ketone functional group, there is no this free hydrogen. So in the case of ketones, there are two alkyl groups. So here is also R. So you see now the most important thing actually is to try to understand the functional group. The aldehyde and the ketones, they are the major functional group in carbohydrates. And the major differences between this aldehyde and ketone functional group is free hydrogen. Like for example, look at it. This is this carbon 1 here, as you can see clearly here. This carbon 1, there is a free hydrogen here, one free hydrogen attached to the carbon. So this is to tell you that this is aldehydes. And if this R is replaced with hydrogen, with alkyl group or with another carbon atom, so it means that it is not longer considered as an aldehyde, rather is ketone functional group. So the defined carbohydrate, it is defined as polyhydroxyaldehyde or ketone. So it means that there must be aldehyde and ketone functional group with many OH. The polyhydroxy means many OH. So they have many OH groups. So these are the two major characteristics of every carbohydrate. Number one, they must have aldehyde or ketone functional group with many OH group. Or maybe if you have a compound that after you hydrolyze it, the word hydrolysis means breaking down. After you break down the compound, maybe at the end it will give you the polyhydroxyaldehyde or ketone. So those compounds, are defined as carbohydrate. Polyhydroxyaldehyde ketones, there must be aldehyde and many OH group, or ketones functional group with many OH group, or any compound that after you hydrolyze it, that is after you break it, it will give you the it will give you such compound. So more importantly, carbohydrates are organic molecules that are made up of three major elements. And these three major elements are carbon, hydrogen, and oxygen. And you can see clearly for the whole of this D-glucose, D-mannose, you see that there are just three elements: the carbon, hydrogen, and oxygen. So the major organic molecules that actually, or the major elements that are found in carbohydrates are these three major elements. So they are one of the four major biomolecules essential for life. Of course, we have four major macromolecules that are essential for life. We have proteins, we have nucleic acids, and then we have lipids. So carbohydrates, lipids, proteins, and nucleic acid are the four major biomolecules that are essential for life. So carbohydrate is one of it, and that is what this statement is trying to talk about. And then, of course, this carbohydrate is served as a primary energy source for every living organism. So, ladies and gentlemen, what I want us to understand is that generally in every living organisms, we need energy. And of course, apart from carbohydrates, the other biomolecules that I mentioned, lipids and proteins, they also produce energy in the body. They also produce energy in the body. But the primary energy source in the body is carbohydrate. But in fact, the energy that is produced by the lipid is even far better than the energy that are generated from carbohydrate. So energy in lipids generated is higher than that of the carbohydrate. But one important thing with carbohydrate, it is the primary source. So if there is carbohydrate, then lipid will not be generated or will not be utilized for energy generation. So that is what we mean with the primary source of energy. So, and then second, this carbohydrates is classified into three main categories. It's classified as monosaccharides, oligosaccharides, and polysaccharides. And we can clearly see here, a carbohydrate is divided into complex carbohydrate and then in simple carbohydrate. So the simple carbohydrate is actually considered as a monosaccharide and disaccharides. But of course, the disaccharide is an example of oligosaccharide because oligosaccharide is a sugar that contain 2 to 15 simple sugars. So disaccharide is 2 simple sugars. So that's why it is called disaccharide. So monosaccharides are the simplest form consisting of a single sugar unit. And this single sugar unit includes glucose, fructose, and galactose. They are examples of single sugar units. So these are the example of single sugar units. We have many of them, but these are the three most examples of monosaccharide. We have glucose, we have fructose, and then we have galactose. So these are actually the three examples of monosaccharide. So they are the simplest sugar. So if you have two of these monosaccharides joined together, then it will give us what we call disaccharide. So it means that disaccharides are generated from the combination of two simple sugars. And then, of course, as we said, oligosaccharide and polysaccharide are composed of multiple sugar units that are linked together by a special types of bond called Glycosidic bonds. So it means that in the case of oligosaccharide, there are multiple single sugar units that join together. So we are going to look at them one after the other, but we should now put this at the back of our mind. Examples of this disaccharide, and you shouldn't forget it. We have maltose, we have lactose, we have sucrose. So these are the examples of simple disaccharides that are actually found in living organisms. So they are disaccharides. And then we have polysaccharides. So polysaccharides, it means that they contain many simple sugar unit, many single sugar units. Like for example, in the saccharide, you can have even 10,000, 20,000, 100,000 simple sugars joined together. And examples of them, we have starch, we have fibers, we have glycogen. So these are the examples of polysaccharides. And of course, this polysaccharide is also divided into two. We have homopolysaccharide and heteropolysaccharide. So because this is actually an introduction to the carbohydrate. So as time goes on, we are going to look at all those examples of polysaccharide. So then now we are going to look at the structure of monosaccharide. So monosaccharide typically have a backbone of carbon atoms with hydroxyl groups. You see, hydroxyl groups. So these are hydroxyl groups, OH group. And then we have many OH group. So we have many OH group. So monosaccharide are simple sugars that constitute the building blocks of more complex forms of sugar. You see, so monosaccharide is a simple sugar, and it is actually the sugars that constitute the building blocks of a more complex forms of sugars like the polysaccharide. So it is actually these monosaccharides that joined together to produce the complex forms of the sugars in the case of polysaccharides. And examples, the common examples of this monosaccharide include glucose, fructose, and galactose. So these sugars, they can actually exist as either in a linear form. Like for example, in this case, they are in a linear form. This is a linear form. And then of course, they can be actually exist in a cyclic forms. So with the cyclic form being the more prevalent in the solution. So most of these monosaccharides, they form a cyclic form in a solution. So they are mostly cyclic in a solution. So these are actually how the monosaccharide can exist. They can be either linear or cyclic, but they are mostly found as cyclic in a solution. So of course, common monosaccharides. So glucose. So we are going to look at these common monosaccharides and their importance. So glucose is a primary energy source, and it is crucial for cellular respiration. So it is the primary form of, so it's a primary energy source. So generally, when we take glucose, it is easily generates energy in our body, and it is crucial for cellular respiration. So cellular respiration is actually when this glucose is breaking down in the presence of oxygen to release energy. So glucose is a primary source of energy. And of course, we have a fructose. So a fructose has been found in many fruits. So it is sweeter than glucose, and it is metabolized differently. So this fructose, it is actually is sweeter than glucose. It's sweeter than glucose, and it's actually found in many fruits. Many fruits, fructose is actually there. And of course, the next one is galactose. So the galactose is a common, or is a component of lactose. And of course, we know that lactose is the sugar that is found in the milk. Which milk? The milk for breastfeeding, the milk that we are taking for tea, like pig and others. So the galactose is a major component of lactose. So the lactose that is found, the sugar that is found in, of course, the sugar that is found in the milk is lactose. And galactose is a major component of the lactose. And of course, look at the structure here and you can see the difference clearly. You see, in the case of galactose, you see that there is a actually, like for example, in the case of galactose, look at the OH here, OH, but look at the fructose and look at the glucose. So these are actually the three examples of or the most common examples of monosaccharides, and they are found, these are the cyclic form of the monosaccharide that are mostly found in a solution. So then the next thing is the formation of disaccharides. So disaccharides are formed by condensation reaction between two monosaccharides. So if you have two monosaccharides and they condense together, they actually produce a disaccharide. And this solution actually results in the formation of a glycosidic bond and the release of a water molecule. So let's look at it here. It is actually a condensation reaction where the OH group of one glucose here react with one hydrogen of the another glucose molecules and they condense. And once they condense, they form a bond, and this is the bond, which is called alpha 1 glycosidic bond. Why is it that it is called alpha 1 glycosidic bond? Is because here, look at it, the OH here is coming from carbon 4, and then the free hydrogen here is coming from carbon 1. So, and it is alpha. The carbon, all these carbons, they are called alpha carbon. So that is why it is called alpha 1 glycosidic bond. So as we are going on, or as we are doing the lecture, don't forget about this. As we are doing this lecture, we are coming to where we are going to differentiate between alpha and beta carbon. So we are coming to that. So you see, for monosaccharides to join to produce a disaccharide, glycosidic bond must be involved. So you see here we have one monosaccharide, that is alpha glucose plus another alpha glucose joined together to produce a maltose with the release of small molecules, which is water. So this is a condensation reaction. So a common disaccharide includes examples of the common disaccharide. We have sucrose, we have lactose, and then we have maltose. So these are the examples of the common disaccharide. So the next thing is actually structure and functions of disaccharide. So let's look at the structure and their function. So sucrose is composed of glucose and a fructose, and it is commonly known as a table sugar. So of course, the sucrose is made of glucose and fructose. So let's look at it. So this is sucrose, and of course this is the glucose. Sorry, this is the fructose, and then this is the glucose. So they join together to produce sucrose by glycosidic bond. And this sucrose is found in table sugar, in sugar cane, etc. And then the next one is lactose. So lactose consists of glucose and galactose. So galactose and glucose are the monomeric sugars that actually join together to produce lactose, and it is actually found in dairy products and also milk for breastfeeding. So then the next one is maltose. So maltose is actually formed by two glucose molecules. So when two glucose molecules, glucose and glucose, join together, they produce a maltose. So maltose, and it is actually produced during starch digestion. So these are the three most common examples of the disaccharide that is found in humans. So the next thing is polysaccharide. So polysaccharide, of course, are large molecules, and it is composed of a long chain of monosaccharide units. So it's when long chain, or it's when long monosaccharide or many monosaccharide unit join together. Like for example, look at it here, 1, 2, 3, 4, 5, 6, continuous. This is, you say, repeated. So it will continue. So actually, it's when many monosaccharide unit join together, that is when we are going to have polysaccharide. So polysaccharide are carbohydrates formed by long chain of repeating unit linked together by glycosidic bonds. So you see all these sugars, one sugar joined with another sugar through the glycosidic bond. Also here through the glycosidic bond through. So it continues like this. So they can be linear or branch. So they can be linear or branch. So like, for example, you can see clearly here, the starch is linear. You see the cellulose is also linear. But look at the glycogen, it formed branch here. It formed branch at carbon 6. It formed branch. So you see, starch and cellulose are linear. Don't forget about this, ladies and gentlemen. Starch and cellulose are linear, while glycogen is branch. So this linear and branching is affecting their properties and functions. So polysaccharides serve various roles, including energy storage and structural support. So this polysaccharide actually is trying to actually have so many roles. Like, for example, if we eat a lot of foods, and of course we have an extra glucose, so that extra glucose will be transported to the liver and store in the form of glycogen. So glycogen is a storage form of energy in human. So when you have excess carbohydrates, so you have excess glucose after digestion. So the remnant glucose will be transported to the liver. It will now assemble itself and convert into glycogen, and the glycogen will store in the liver. And after we exhausted our energy, then the glycogen will be mobilized, will be released, and then break down to release glucose into the blood for the maintenance of blood glucose level as well as the energy production in the body. And of course, we have some of the carbohydrates that actually give structural support. Like, for example, cellulose is a main carbohydrate that is found in plant, and it is, of course, very important in maintaining the structural integrity of the cell wall in plant. And another example, we have hyaluronic acid. Hyaluronic acid is also examples of polysaccharide. It is found on the egg of female. And of course, fertilization will not even take place without even breaking down of that hyaluronic acid, which is a polysaccharide. And of course, our sperm is also rich in an enzyme called hyaluronidase, and it is the of course that hyaluronidase that break down hyaluronic acid that allow the sperm to get access to the nucleus of the egg cell. So this hyaluronic acid is polysaccharide that actually trying to protect the integrity of the egg of female. So examples here of let's actually start with the starch, which is one of the polysaccharide. So starch is the storage polysaccharide that is found in plant. So it is found in plant. Then it consisting of amylose is divided into two. So it consists of amylose and amylopectin. So we should get this, and we also explain the difference between amylose and amylopectin in our next lecture. So this starch is served as the energy reserve that can be break down in glucose when needed. So that is actually starch. So we know that we as human, we take most of our carbohydrate in the form of starch. So we'll break it. After we break it down, it releases glucose. So we have amylose and amylopectin. Amylose is linear and amylopectin is linear and branch. So the next thing is actually starch is commonly found in foods such as potatoes, rice, and corn. So these are the main sources of starch. So then the next thing is glycogen. So glycogen is a storage form of glucose in animals, and it is highly branched. So you see glycogen, it is highly branched, and it is the storage form of glucose in animals, and I explained that. And it is also primarily stored in the liver. It's stored in the liver and muscles. Yes, apart from the liver, glycogen is also stored in the muscles. But the glycogen stored in the muscles is mainly made for the muscles to utilize it for energy. So, and also providing a rapid energy source. Is a rapid source of energy. So once there is no energy, then we have the storage we have the stored glycogen, which will release the glucose for energy generation. So the glycogen in the liver will release glucose to maintain blood glucose level and energy generation by other cells of the body. While the glycogen in the muscles is meant for the muscles for energy generation. So glycogen can quickly mobilize during period of increased energy demand. So if our body that is in need of energy, if there is an increase in energy demand, of course the glycogen will be mobilized, break down, and utilized for energy generation. So then the next one is cellulose. So a cellulose is a structural polysaccharide that is found in cell walls of plant. So it is found in cell walls of plant. I explained that. And it is composed of linear chain of glucose units linked by beta-1,4 glycosidic one. So you see, in this case, ladies and gentlemen, this one is beta glucose. Why is it that it is beta glucose? Is because of the OH group here. So as I said, we are common, we'll explain the concept of alpha and beta. So it is from a beta glycosidic one. So human cannot digest cellulose because we don't have the enzymes that digest cellulose in our body. So we don't have the enzymes that digest cellulose in our body. That is why we cannot digest it. Although we have some intestinal microbial flora that are rich in cellulose, and of course they are the one that help us to digest it. But it is important for dietary fiber and gut health. That is microbial organism. So the next one is chitin. So chitin is a structural polysaccharide that is found in the exoskeletons of arthropods and fungi. So this chitin is also another example of polysaccharide, and it is found in the exoskeletons of arthropod and fungi. Arthropod is insect, so it's found in like mosquito, like grains, weevils, fungi. This chitin is highly rich there. So they have it, and it is actually the carbohydrate that give them structural supports. And then it is composed of, so you see the repeating unit of chitin here is N-acetyl glucosamine. So it is N-acetyl glucosamine unit that linked together. Like in the other case, most of the repeating unit of cellulose, glycogen, and starch is glucose. That is the repeating unit. But in the case of chitin, the repeating unit is N-acetyl glucosamine. So chitin, it actually provides rigidity and protections to organisms that possess it, like mosquito, other insect, and fungi. So then let's actually look at the glycosidic one. So glycosidic bonds are the linkage formed between monosaccharides and carbohydrate. So the type of glycosidic bond, it can be alpha or beta. Which influences this alpha and beta influences the properties and digestibility of the carbohydrate. So enzymes are required to break down glycosidic bond during digestion. So of course, if we take our carbohydrate, so the digestion that is taken in our body is using enzymes. And the major function of the enzyme is to break down the glycosidic form. So if we doesn't have the enzyme that will digest glycosidic one, like alpha, beta glycosidic, alpha 1,4 glycosidic one, it means that we cannot digest it, we as an animal. So what are the carbohydrate function in our body? So carbohydrates serve as the energy sources. We explain this, and it also provides structural support, and it's also important as a signaling molecules. When you say signaling molecule, it means that there are molecules that are released to send message between one cell to another cells, or to commit to it help cellular communication. That is help one cells to communicate with another cells using the carbohydrate. So that is why it's called signaling molecules. So a signaling molecules is a molecules that help in cellular communication, cellular interaction, and so many other things. So they play a crucial role in cellular recognition. So it is very important, this carbohydrate, they play a role in cellular recognition. Like for example, how one cell recognizes its own cell, how one cell recognize a particular cell is not actually a human cell. Like, for example, how do we react to pathogens? How do we react to microorganisms is actually through these signaling molecules. So these signaling molecules are the molecules that if a virus infects a particular cell, so it is this carbohydrate that recognize that this is not my own cell. It is a viral cells and it needs a proper action for the microorganisms to be eliminated. Communication also at the same time. Like, for example, how sperm cell and egg cell communicate is through this kind of biomolecules like carbohydrate biomolecules or glycoproteins. They are the molecules that, like, for example, if after fertilization, sperm, after ejaculate, after you release sperm, it's actually transport from one part of the woman to the fallopian tube. So how sperm cell recognize that this is an egg cell that actually I should or the sperm cell should recognize it and actually fertilize it. So how sperm cell and egg cell recognize each other for the fertilization to take place is through the cellular recognition and communication. So carbohydrate play a better role. So it means that it is a carbohydrate that help that help one cells to recognize one another, that help one cells to communicate with another cell. Like, for example, if let's say there is, let's say 1,000 cells or 1 million cells in the liver. So if a liver cells is infected with the virus, so how the infected cells communicate with the neighboring cells is through carbohydrates, because the infected cells uh, will now send signals or will send message to the neighboring cells that this is a virus that infected my own cells, so you should take care. So those cells will now prepare themselves. Even if those, or even if that organism is trying to infect them, then they'll protect themselves. So glycoproteins and glycolipid are the examples of polysaccharide that involve in cell signaling. So what is cell signaling is the communication. How one cell sends a signal, a message to another cells. So these are the examples of the carbohydrate. As you said, function, it quickly generate energy, it actually store energy, it also give structure. And examples, we have sugar, starch, and cellulose. So ladies and gentlemen, this is where we are going to stop for today's lecture. It is just an introduction, and we are going to look at the concept of carbohydrate in more details in our subsequent lecture. And I hope you find this lecture very interesting. Thank you very much.